Am I audible? Am I audible? Am I audible? Hello guys, how are you? Hello guys, am I audible? You guys can hear me. I'm live. Are you ready for the questions, the PYQs today? Shall we start or should we wait for like another minute or so? Chalo, let's wait for another 1 minute and then we'll start. Alright, so it's time to start now. Um, Before starting on with the session, I'll just briefly introduce myself. My name is Aarushi Anand. My name is Aarushi Anand, and I am I'm pursuing BSc Zoology from Hindu College, and I appeared in CUET exam 2023, and I got 100 percentile in bio, psychology, and English, and 99.8 around something that I got in physics and chemistry. Okay. Before moving further, there's a question in the chat कि BSc zoology के बाद आगे career options क्या होते हैं? So there are a lot of career options. ठीक है. Personally, what I want to do is research. So you can do research in any of the related fields like neuroscience, biotechnology, biochemistry, anything that interests you. Other than that, you can if you're interested in going into teaching jobs or uh, professor jobs, so that you can do. You can be a researcher, you can be a professor, you can also change your streams. After that, you can be a psychologist after that. So like that, yeah, there are options in after BSc zoology. And if you maintain a good score throughout the time that you're doing your graduation, there are chances that you can get like scholarships and everything, and then you can go for research out of country. So then that will help you if you are planning to do research. Then yep, exam factors का mock test series के बारे में I've already told you that this is available for the subjects that are mentioned on your screens. है ना? So mock test is important because you need practice. You need to strategize your CUET paper, the attempt of the CUET paper. You need more and more practice. जितना आप practice करोगे उतना आपको ऐसा नहीं लगेगा that you are doing CUET for the first time. Like जब paper देने का time आएगा, right? And the more you practice, the more uh, the more chances are there that you will ace your exam. And about the crash course, it's the revision. So revision and practice are the two most important things that is needed, that is required right now. We are doing PYQs that so that you know how the pattern is gonna be. But since you cannot predict the questions because it has been like only 2 years that CUET has been there, and you never know how what difficulty level is there the next year. Sorry, this year, and what kind of questions are going to come. So you need practice and you need revision. So you can check out ExamFactor's website, and you can avail the offers and avail scholarships as well. So yes, coming on to the questions part. So let's start questions. Before starting on, have you guys solved the previous questions? Did you see the previous videos, or like were you there when we were discussing the PYQs before? Are you guys ready? Should we start? Are you guys ready again? Should we start? Okay, so let's start with the first question on your screens. Mature mRNA is a fully processed 1 hnRNA, snRNA, 28 sRNA, and 5 srRNA. So you have to tell me mature RNA is a fully processed what? Also, mRNA is what? mRNA is M stands for messenger RNA, right? So it's basically a messenger that helps in the uh, transcription and translation from DNA to DNA to RNA, then RNA to proteins. एग्जाम डेट ईच सब्जेक्ट की अलग अलग है, सो कैसे पैटर्न होता है एग्जाम का? सो ऑब्वियसली एग्जाम डेट अगर अलग अलग है तो क्वेश्चन चेंज हो जाते हैं, बट पैटर्न सेम रहता है। 50 क्वेश्चन है तो यू ओनली नीड टू अटेम्प्ट 40 आउट ऑफ देम। 8 you can leave, sorry 10 you can leave, and you have to strategize your approach while giving CUET. Acha, now first question on your screens. Mature mRNA is a fully processed what? hnRNA, snRNA, 28sRNA, and 5srRNA. This is like very very easy, very very easy. I'm waiting for your response. Okay, so I'll only tell the first option is the correct option. Mature mRNA is a fully processed hnRNA. ठीक है? um, आंसर भी देख लेते हैं एक बार। ठीक है, so option 1 is correct. As I said, mature mRNA is completely processed mRNA molecule in the cell of the eukaryotes. The mRNA is a type of RNA involved in protein synthesis. It is synthesized based on a DNA template on the nucleus, और वो DNA template को क्या कहते हैं? I mean DNA template से RNA बनने को क्या कहते हैं? हाँ, MCQ से होता है एग्जाम का पैटर्न। I mean MCQ से होता है। अभी हम MCQ से सॉल्व कर रहे हैं ना। Okay, now the नहीं OMR filling नहीं होता. It's a CBT based model. Like you have to do the like attempt the paper on a computer that would be there आपकी center में. It's CBT, that is computer based test. Anyway, second question. Which forest also known as the lungs of the planet Earth? 1. Amazon rainforest, 2. Rainforest of Northeast India, 3. Taiga forest, and 4. Tundra forest. This is like the easiest question. You've been doing this since what, 7th class? So just giving you 10 seconds इसका आंसर बताने के लिए. So the answer for this is Amazon rainforest. So Amazon rainforest is also known as the lungs of the planet Earth. Why? Because they are responsible for the production of like around 20% of the Earth's oxygen. They remove a large amount of carbon dioxide from the air. ठीक है, so the answer is Amazon rainforest. ठीक है, now question number 3. Inbreeding is carried out in animal husbandry because it first increase hybrid vigor, improves the breed, increases heterozygosity, and increase homozygosity. Giving you 30 seconds to think about this question. इसका आंसर बताओ. इसका आंसर I also need a while, like why, an explanation. ठीक है, तो अगर हम इन ब्रीडिंग करा रहे हैं, इन ब्रीडिंग का मतलब क्या होता है? Inbreeding is breeding, like carrying out a cross within the same breed. अगर हम सेम ब्रीड के अंदर क्रॉस करवाएंगे, दैट सेम ब्रीड मीन सेम ट्रेड्स, अगर सेम ट्रेड्स जनरेशन टू जनरेशन, लाइक 4 to 6 जनरेशन हम सेम ब्रीडिंग करवाएं, तो वो हेट्रोजाइगोसिटी तो बिल्कुल भी नहीं बढ़ाएगा, है ना? हाइब्रिड भी नहीं होगा बिकॉज इन ब्रीडिंग हो रही है, improves the breed भी नहीं होगा. So the only answer we're left with increasing homozygosity. So the answer is homozygosity, and I'll tell you why. क्योंकि हमें क्या चाहिए होती है? प्योर ब्रीड लाइन्स चाहिए होती है, प्योर सेम प्योर लाइन एनिमल चाहिए होता है, ताकि मतलब अगर एक हाईली रिप्रोड्यूसिंग फीमेल है और एक हाईली स्ट्रॉन्ग मेल है, तो हम उनको क्रॉस करवाएं, फिर उनकी ब्रीडिंग करवाते रहे, इन ब्रीडिंग करवाते रहे, ताकि वो प्योर लाइन बन जाए एट द एंड ऑफ द डे. नाउ फोर्थ क्वेश्चन ऑन योर स्क्रीन, व्हाट इज नोन एज द टेरर ऑफ बंगाल? आज के क्वेश्चंस तो इजी आ रहे हैं ना. व्हाट इज नोन एज द टेरर ऑफ बंगाल? Okay, so the terror of Bengal is water hyacinth, right? Option number 3. It is also known as Ecornia, right? So why is it known as terror of Bengal? Because it is an exotic species. It is not an indigenous species. And जब इसको हम लाए और हमने grow किया, so it was an aquatic organism, and it cut down all the light and all the oxygen supply of oxygen that went beneath the वाटर, और ये एलियन स्पीशीज इन्वेजन का एग्जाम्पल है. इट्स एन एग्जाम्पल ऑफ एलियन स्पीशीज इन विच नेटिव स्पीशीज या इंडिजिनस स्पीशीज वर इन थ्रेट बिकॉज ऑफ दिस इकॉर्निया और वाटर हाइसेंथ और टेरर ऑफ बंगाल. Alright, so fifth question on your screens. Threatened animals and plants are taken out from their natural habitat and placed in special settings, protected and given special care. Is this process is known as what type of conservation? Is it conservation in national park, conservation in biosphere, in situ conservation, and ex situ conservation? You guys tell me the answer. Till the time I'll just bring my charger quickly. Yes, so what is the answer for this? So इसका answer is pretty simple. It's an example of XC2 conservation. So in XC2 conservation, what we do that we take animals or plants, threatened animals or plants, and we take them out of their natural habitats and we place them in special settings. और इसके examples क्या होते हैं? Gene banks या फिर gene banks, seed banks, or zoo or zoological gardens, botanical gardens, right? And बाकी ये जो biosphere reserves वगैरह दिया है, that is in situ. Biosphere reserve is an in situ conservation. Now question number 6 on your screens. इसमें शायद आपको टाइम लगे ये वाला सॉल्व करने में, तो I'll give you 1 minute to solve this. Sorry. चलो इस क्वेश्चन को सॉल्व करते हैं. 6 says sequence of the following steps in the formation of female gametophyte. Female gametophyte of flowering plants. Sequence बतानी है. A says cell walls are laid down. ठीक है. Okay. Formation of 7 cells with 8 nuclei. Okay. Meiosis in megaspores. Mother cell and formation of a megaspore tetrad. Functional megaspore undergoes 3 mitotic division resulting in form of 8 nuclei. Okay. तो सबसे लास्ट में तो ये होगा B है ना. So we'll have to see B किस में है लास्ट में, 3 में और 1 में. 1 का first is cell wall is laid down, which is incorrect. 3 का is CDAB. So C is correct. C has to be the first one. Then D, because mitotic divisions होगी, तो मियोसिस होगा, उसके बाद माइटोटिक डिविजन्स होंगी, मतलब एक टेट्राड बनेगा और टेट्राड में से टेट्राड में से only one of them would be functional megaspore, and it's known as monosporic development also, right? तो उसके बाद मियोसिस होगी, ठीक है, आपके वो न्यूक्लियर सेंट्रल सेल्स बनेगा, देन एंटीपोडल सेल्स बनेंगे, एंड न्यूसेलस बनेगा, राइट? सॉरी, न्यूसेलस बाहर होगा, फिलीफॉर्म अपैरेटस बनेंगे, वो सब बनेगा, ठीक है। देन सेल वॉल्स वुड बी लेड डाउन जब न्यूक्लियाई हो गया, क्योंकि जो फ्री न्यूक्लियर डिवीजन होती है, देन सेल वॉल बनेगा। उसके बाद आपका सेल्स, एक सेकेंड, उसके बाद 7 सेल्स एंड 8 न्यूक्लियाई वाला सेल आपका बन जाएगा, सॉरी, फीमेल गैमेटोफाइट बन जाएगा। तो आंसर इज 3, आंसर इज 3। So, seventh question on your screens. It says, in which process unusual nucleotide, which is methyl guanosine triphosphate, is added to the 5' end of hnRNA? Again, a very easy question. इसका सिर्फ एक ही आंसर हो सकता है, बताओ। Right, so what is the answer? The question says, in which of the process unusual nucleotide methyl guanosine triphosphate is added to the 5' end of hnRNA? तो hnRNA से क्या बनता है? mRNA, right? फंक्शनल क्या होता है, mRNA या hnRNA? mRNA, right? तो उसके बाद ऐसा क्या होता है hnRNA में जो कि mRNA बन जाता है? There are two processes that happens, which is splicing and capping. Splicing and capping, right? But is what? Splicing is आपकी जो functional unit नहीं है उसको हटाना. So it can only be one thing, which is capping. So the answer is option number 2, capping. Option number 2, capping. ठीक है, now question number 8 on your screens. Select the incorrect statements given below. Methane and carbon dioxide are greenhouse gases. The Montreal Protocol is associated with the control of emission of ozone depleting substances. Use of incinerators is not crucial to dispose of hospital waste. Dobson unit is used to measure water quality. Now, what is the answer? Right. So, methane and carbon dioxide are greenhouse gas. Yes, they are the greenhouse gases. The Montreal Protocol is associated with the control of emission of ozone depleting substances. Again, yes, correct. Use of incinerator is not crucial to dispose of hospital waste. Incorrect, because use of incinerator is crucial to dispose of hospital waste. Dobson unit is used to measure water quality. Quality. Dobson unit is measure the thickness of the ozone layer. So the incorrect statements are C and D. C and D. That makes it your option number 2. So option number 2 is the correct option. इसमें तो कुछ समझने वाला नहीं है. Now question number 9 on your screens. Question number 9. Yeah. Biofertilizers are the organisms that enrich the nutrient quality of the soil. Which of the following is not a source of biofertilizer? Which of the following is not the source of biofertilizer? This we like discussed microbes and human welfare yesterday, so I am as I hope that you guys know the answer for this question. Also, आज हम strategies वाला chapter करेंगे and we will also uh, we will also make tips and tricks to remember that strategies वाला chapter. So, okay, bacteria. क्या बैक्टीरिया न्यूट्रिएंट क्वालिटी इंक्रीज करता है? ज़रूर करता है. हमारे पास एसिटोबैक्टर है, हमारे पास एसोस्पाइरलम है, ठीक है. ये सारे जो बैक्टीरियाज है, फ्री लिविंग बैक्टीरियाज है, फ्री लिविंग नाइट्रोजन फिक्सिंग बैक्टीरियाज है ना. अगर हम साइनोबैक्टीरिया की बात करें, साइनोबैक्टीरिया करता है, हाँ बिल्कुल करता है. हमारे पास नॉस्टॉक है, हमारे पास एनाबाइना है, हमारे पास ऑसिलेटोरिया है. फंगाई की बात करें तो फंगस करता है क्या? फंगस में हमारे पास ग्लोमिस है ना, राइट. ये सारे हमारे क्या है? नाइट्रोजन फिक्सिंग. ग्लोमस क्या है हमारा? ग्लोमस इज फॉर phosphorus and this is for nitrogen again. Now what is baculovirus? ये हमने कल ही पढ़ा था that baculovirus, they do not act as a fertilizer but they act as a pest controlling agent. Pest controlling viruses basically. ठीक है, they are very insect group specific and they help in IPM, which is IPM is integrated pest management, है ना, and they do not affect the uh, non-targeted insects. ठीक है. Now question number 4. वो क्या? Right, the answer for this. Answer is answer is 3. ठीक है. Answer is 3. Now question number 10. Identify the gene which is effective against corn borer. मैंने इसकी ट्रिक अगेन बताई थी. वैसे कौन बोरर होता है, राइट. इसकी एक ट्रिक है. देखो ये आंसर तो बिल्कुल भी नहीं है, तो इसको हम नो कर देते हैं. अभी क्राई LAC ये एक्चुअली आई होना चाहिए, एंड क्राई अगेन ये आई होना चाहिए. क्राई 1AC, क्राई 1AB, क्राई 2AB. ठीक है. एंड ये विच इज नॉट इफेक्टिव होना चाहिए. एक सेकेंड में चेक कराती हूँ. ओके सॉरी, माय बैड. हाँ, तो इसकी मैंने ट्रिक ये बताई थी कि आप 1 को, ये जो है, इसको आप कंसिडर करो लाइक क्राई 1 AC जब हम बोलते हैं इसको कंसिडर करो 1, A को कंसिडर करो 1, C को कंसिडर करो 3. तो 1 + A + C इज हम 5. ठीक है. कॉर्न में कितने लेटर्स हैं? 4. तो हमें ये कुछ ऐसा कॉम्बिनेशन ढूंढना है जिसका 4 हो. तो 1 + A + B एंड 2 + A + B. तो 2 + 1 + 2 इज अगेन 5. तो सही आंसर हमारा होता है ये. आई मीन ये एक बहुत ख़राब ट्रिक है, बट दिस इज समथिंग इन विच यू कैन रिमेंबर. ठीक है. मैं अगर लिखूँ 1 प्लस A is equal to 1 again plus B is equal to 2, which is equals to 4, and corn is equal to 4. So this becomes the right answer. I mean ये बहुत खराब ट्रिक है, I know again, but याद रहेगा एटलीस्ट. सो कोई क्वेश्चन में डाउट है? एक बार रिवाइज कर लेते हैं. ठीक है. सो दीज 2 वर द करेक्ट स्टेटमेंट्स, है ना. दीज 2 वर करेक्ट थी ये दोनों, and ये दोनों were wrong, which then our answer was 2. C and D wrong. ठीक है. ये वाला भी हो गया. कैपिंग was the answer. I hope it's clear. ठीक है. ये भी then sequence you were able to find out. Then XC2 conservation, it's like the easiest question ever. ठीक है. Then water hyacinth also you know. Then हम आज यही चैप्टर करते हैं. चलो. इंक्रीज होमोजाइगोसिटी, what happens, why it happens, ये भी पता चल जाएगा. And obviously Amazon rainforest are the lungs of the world, planet Earth. This is very given. And H and RNA was the right answer. It's heterogeneous. What is the full form of H and RNA? बताओगे तो अच्छा लगेगा. What is the full form of H and RNA? मैं बताती हूँ चलो. HNRNA is heterogeneous nuclear RNA. Heterogeneous nuclear RNA and it is formed immediately after transcription. फिर transcription में हम क्या करते हैं, हम जो उसके exons that is the non coding sequence होती हैं उसको splicing करते हैं and methyloguanosine की capping होती है and then what is ready, mRNA is ready for translation. So these were the questions that we did today, and I'll just give you 2 minutes break. मतलब I'll give you 2 minutes break, या you guys give me 2 minutes break, and then उसके बाद we'll start with the strategies chapter, right? मतलब I'll just need 2 minutes to open the PDF for strategies. So give me 2 minutes, please.